Swayam Prabha. Digital India. Educated India.
In previous lectures concerning centrifugation, we have discussed about the basic principle of centrifugation techniques, and also we have discussed about different types of centrifuges. In basic principle, as if you could recall, there are many factors on which the sedimentation rate depends. One is applied centrifugal field, and it is expressed in terms of most of the time relative centrifugal field, that is multiples of the gravitational field or G force we call it. Also, like we have seen, there are other factors which will determine the sedimentation rate, like the size of the particle, shape, density. In shape, it is like whether a particle is spherical and non-hydrated, or it is aspherical and hydrated. Also, the viscosity of the medium and the density of the medium will also play an important role in determining the rate of sedimentation. So, all these factors where you have angular velocity or it could be expressed in terms of revolutions per minute, and it is being expressed in terms of relative centrifugal field, and sedimentation rate in terms of the acceleration due to the centrifugal force and the frictional force which is generated due to the acceleration of the particle in a medium under centrifugal field. All these factors play an important role, and we have discussed about them. Sedimentation rate or sedimentation velocity could be expressed in terms of sedimentation coefficient also, that is velocity per unit field, and it is expressed in terms of Swedberg units, that is 1 Swedberg unit is 10 raised to the power minus 13 seconds. So, higher the Swedberg unit, then higher will be the sedimentation rate. Then we have discussed about different types of centrifuges, and if you could recall in last lecture, we discussed from very small benchtop centrifuge to high capacity refrigerated centrifuge, high speed refrigerated centrifuge, and the analytical ultracentrifuges. Also, we have discussed about in detail about the analytical ultracentrifuges. Now, in this lecture, we are going to discuss about the separation methods which are based on preparative ultracentrifuges, and you can say these are preparative methods where you are aiming to get the material for further biochemical investigation. In analytical ultracentrifuge, in contrast, it was for determining the sedimentation behavior and other aspects could be done because of an optical system which is attached to the analytical ultracentrifugation technique instrument. So, if you could recall, analytical ultracentrifuge is mostly utilizes, or samples which are taken are either purified or at least partially purified for characterization. So, let us start with certain or some separation methods which are widely used in preparative ultracentrifuges, and also like it is the you can say high speed centrifuges also. So, we will start with we are going to discuss in this section. One is differential centrifugation, which is a very common method of separation, particularly subcellular organelles or cell components. Then we are going to discuss about density gradient centrifugation, where we are going to discuss about two methods, rate zonal centrifugation and isopenic or isodensity centrifugation method. So, let us start with differential centrifugation method. Now, differential centrifugation method is the most common technique and it is called like most common technique and is widely used for separation of particles from a liquid medium or to separate particles of different masses into separate fractions into the pellet and supernatant. Now, separation is achieved primarily based on differences in the sedimentation rate of the biological particles of different size and density. Now, here the liquid medium is a simple particular density material. Here mostly the separation will take place on the basis of mass of the particle, or you can say size and density in terms of how it sediments, what is the rate of sedimentation of that biological particle. So, various fractions or various components of in a particular sample will be separated on the basis that of their differential rate of sedimentation. Now, obviously, particles with large size will sediment much faster as compared to small size particles. So, larger particles will be pelleting first and will require lower centrifugal force and medium sized particle will then follow up with a higher centrifugal force, and then finally, a small sized particles could be pelleted will require much higher centrifugal force. So, for if you consider for same mass particle, but having different sizes, the centrifugation will mainly sediment higher density particles. So, if certain particle have same mass, but densities are differing, then higher density particle will be sedimented first. Particles having similar bending densities, like many times you have these subcellular organelles which have densities in the range of say 1.1 to 1.3 gram per centimeter cube in sucrose solution. They could be separated if there is a tenfold difference in their mass. So, there could be one way could be that they differ, they might be of same density, but they differ in mass, they could be separated like we will be discussing red zonal one, or they differ in densities actually. So, that they could be separated on the basis of their differing density. Now, commonly used like in simple pelleting, like you obtain partially pure preparations of subcellular organelles and macromolecules. We will be discussing this that how when you start differential centrifugation and your material is homogeneously distributed across the tube or the tube in which you are doing the sedimentation, you end up getting not completely pure, but partially pure preparations of different materials or components like organelles, macromolecules of subcellular organelles. And for the study of subcellular organelles or tissues or cells, they are how do we go about when you have to say you would like to study different subcellular organelles from a tissue or cells. The first thing to be done is that you have to disrupt the cells, or you have to release the internal components into the solution, and that is done by disruption. So, the crude is disrupted cell mixture which is taken. Now, it could be done by different methods as we have discussed earlier. It could be like say a crude way of breaking the cells, for example, freezing and thawing, or it could be done by sonication or French press applications, or through enzymatic like enzymatic method could be utilized for releasing subcellular organelles or breaking the cell wall. Now, once this is done, then crude disrupted cell mixture will be referred to as homogenate. which contains all these things in a solution in a homogeneously distributed here. So, during centrifugation of a cell homogenate which you have taken after disruption of cell, you will take them in the tube and then sedimentation starts. Now, in differential centrifugation, like I said, it takes advantage of the different sedimentation rates of various components of the homogenate. So, what is done is that the homogenate or the differential centrifugation is done in steps, like stepwise you keep on increasing the the centrifugal force, so that different particles sediment at different centrifugal force or different revolutions per minute. So, when you have one your centrifuge for a particular time, one particular time at particular centrifugal force, then one component or one particle component will settle down, which will be larger component. And then as you increase the centrifugal force by increasing revolutions per minute, then what you get is the smaller components will start pelleting in the order of their size or mass, you can say. So, here larger particles like a sediment faster than smaller ones, and this is the basis for obtaining crude organelles fractions by differential centrifugation. So, tissue homogenate is divided centrifugally into number of fractions by increasing the applied centrifugal field. Now, a cell homogenate can be centrifuged at a series of progressively higher g force and times to generate pellets of partially purified organelle. As you increase the g force and you start with certain g force, this could be if you know about the components, that is they will pellet at particular g force, then you can start with larger one which will be largest component which will pellet at particular g force. You can start with that g force, and also time depending on what tubes or what rotors you are taking, and if you know that, then times could be set, and then you could start centrifugation, and so you will get pellet and supernatant. Now, again what you will do is pellet is taken and supernatant can again be taken for higher g force, and again something pellets smaller molecule, and likewise you can go on to sediment all the components of a homogenate in a series by increasing the centrifugal field. Now, any type of particle present in homogenate may be found in pellet or supernatant or both fractions, depending upon speed and time of centrifugation and size and density of the particle. So, this will be I will show you in a while. Now, initially all particles of the homogenate are homogeneously distributed throughout the tube. So, during centrifugation particles move down the centrifugal tube at their sedimentation rates and start to form a pellet on the bottom of the centrifuge. So, what happens here, when a cell homogenate is centrifuged say at a very low speed, say 1000 x. So, remember in these preparative ultracentrifuges techniques, the ultracentrifugation or very high speeds are used only for a certain components to be pelleted. So, when a cell homogenate is centrifuged at a very low speed, say 1000 g force for say 10 minutes, then unbroken cells and heavy nuclei pellets to the bottom of the tube. So, what you get? You get a pellet and supernatant. Now, the supernatant can be further centrifuged at 10,000 g or any, like you have to choose that for say 20 minutes to pellet certain subcellular organelles of intermediate velocities such as mitochondria, lysosomes or other subcellular fractions. Now, some of these sedimenting organelles are obtained in partial purity and typically contaminated with other particles. So, why they are contaminated? Let me show you on your screen this thing. All right. So, what what is the homogenate cell homogenate is taken in a tube, and this is distributed all over, all right, and this will contain different size fraction. This will, there is one fraction here, this is one another fraction, this could be another fraction, this could be another fraction, and they are distributed all over actually. So, what you have is these are distributed all over. Now, when you apply a particular centrifugal field, for example, say you apply a centrifugal field of say 1000 G, then certain or larger component will settle, like we said, cell debris and other very heavy nuclei could be settling. So, what you will get is you will get a pellet here, depending on how what kind of rotor you are using, pellet will be formed accordingly, and you will get. So, pellet will contain what? Pellet will contain say the biggest one molecule. Now, problem is here why you get partial purity, and you will also get these things which are again distributed all over actually. So, now why you get partial purity? When centrifugal field is applied, then different molecules will sediment at different rates, but these molecules are homogeneously distributed all over the tube. So, a molecule which is here will experience a particular force, a molecule which is here, these are the molecules which are already present at the bottom, they will also experience certain force. So, what will happen is this molecule which is larger molecule will travel down here, but these molecules which are at the bottom only, they will also travel down here, and so what you get is you get a pellet which might contain major fraction portion of the larger molecule, but it will also contain it will also contain other fractions or other components, and this is because they are distributed across, and the one which is very close to the bottom will also sediment in here. So, unless you layer on top of here, this particular problem will be there. So, now what is happening? So, what you have to do to get purify, what is done is you take the supernatant, in next tube you can decant it, and again the centrifugation will be done at higher G force. So, you will do it at higher G force, like say for 10,000 G force or whatever it is. Likewise, you will take this pellet also, and you will again homogenize in a particular suitable solvent solution or medium we can say, and this is again this pellet is which is again which will contain more of these, but it will also contain other smaller ones. Again it will be centrifuged, and this process will be done many times to get the pure fraction. So, partial you can achieve a reasonable purity by going through these steps of repeating the centrifugation at that speed by taking the pellet and then again doing it many times, so that you can get the pure fraction. So, what is done is that centrifugation is continued long enough to pellet all the largest class of particle, and the resulting supernatant is centrifuged at higher speed to separate medium sized particles. Like I said, repeated washing of the pellet by resuspending it in isotonic solutions and repelleting may result in removal of contaminants that are smaller in size. So, you could do it, you can purify to a large extent a particular component by resuspending the pellet in a particular solvent, and then again going through many cycles or at least a few cycles of washing the pellet and again sedimenting it, and then finally you get quite reasonable purified material. So, the separation here achieved may be improved by this particular procedure, and recentrifugation and pelleting and recentrifugation this could be done. So, you can obtain partially purified organelles by differential centrifugation, and it serves as a preliminary step of further purification using other types of centrifugal separation technique. like we will be discussing density gradient separation. So, though you get a partially impure fraction here, but this is a very useful technique or very much used technique for various applications. Uh, all right, let us pay attention whatever we have said, let us see how this is done in here. So, if you see this figure on your screen, what you have is you have you have a tissue homogenization, and remember you have to disrupt the cell by any technique, and then components are released into the solution and they are called homogenate. So, tissue homogenate is obtained or cell homogenate is obtained here, and you can see there are a lot of different components of different sizes, they are equally distributed or homogeneously distributed all over the tube. Now, what you do, first thing is you start with very low speed or G force and you centrifuge at a particular time, say 10 minutes. What will happen once you centrifuge this for 10 minutes? The largest one, these triangles settle down here. So, the pellet contains whole cells, nuclei, cytoskeletons, plasma membrane and other things are there, which pellets in here. Now, again like I said, this pellet is not completely pure or does not constitute only the largest one, there will be smaller ones also. So, the resuspending and again centrifugation, recentrifugation couple of times is required. Now, what is done? So, pellet and supernatant are removed here and then this supernatant is taken in a fresh tube and then again centrifugation is performed. Now, at this time you will go for higher centrifugation, like here it is 20,000 G force for 20 minutes, or it could be like you can go in little smaller steps, like you can start with like 10,000 G or so, but this is just to understand the whole concept. So, what will happen? It will pellet certain other subcellular organelles like say mitochondria or lysosomes or peroxisomes, these will be sedimented. So, next bigger, like this was the largest one and then medium sized particles are being pelleted here. So, again you will get a pellet, this pellet will be partially pure or not completely pure. So, again it has to go through resuspension and recentrifugation and to get the reasonable purified sample. Now, supernatant here will be again taken in a separate tube and then again centrifuged at much higher like say 80,000 G here for 1 hour. Now, you can see that for larger molecules you require low G force as well as less time, but as you go through these steps the smaller molecules require higher G force as well as you require more time for centrifugation. So, again you get pellets which contains microsomes, fragments of ER, small vesicles etcetera, and again this could be purified. The supernatant again will be taken and again done, again it will be recentrifuged, supernatant will be centrifuged at much higher say 100,000 G or force. So, what you will get, pellet will contain ribosomes, macromolecules and other things. Supernatant still might contain certain small soluble proteins which might not pellet as such. So, this is how differential centrifugation is done in steps, and lot of things could be separated on the basis of their rate of sedimentation, which is dependent on the centrifugal applied centrifugal field and their mass. So, this is how centrifugation is done in differential centrifugation. Now, this is very simple way of showing this, that you have a centrifugal field in this direction and as you go along you get different sized fragments separated and could be purified as we go along in here. So, first is low speed, then faster speed, higher speed and highest speed, that is the sequence of events which occur in the centrifugation method, this centrifugation method. All right. So, to summarize, differential gradient centrifugation takes advantage of the differential sedimentation rate or the differences in applied centrifugal field for sedimentation in a particular time. Now, different components, larger, medium sized and smaller components will be separated by gradually or stepwise increasing the centrifugal applied centrifugal field and for different times, and So, you will separate large molecules first, which will sediment first, then medium sized, and then finally, smaller size molecules will be sedimenting at much higher applied centrifugal field. All right. So, we will move on to the next method, that is density gradient centrifugation. Now, density gradient centrifugation is the preferred method to purify subcellular organelles and macromolecules. It is pretty good and gives can purify lot of different types of subcellular organelles and macromolecules like proteins. Now, density gradients can be generated by placing layer after layer of gradient media, such as sucrose in a tube with the heaviest layer at the bottom and the lightest at the top. In either, this could be a discontinuous like stepwise gradient, or it could be a continuous gradient. The cell fraction to be separated, or the particular molecule or material to be separated, is placed on top of the layer and centrifuged. In density gradient centrifugation, what is done is that you can very well separate subcellular organelles, and what is done is, like I said, there needs to be formation of a gradient. So, gradient can be formed. Their gradient is in, it is a increasing like concentration, it is a increasing concentration of the gradient material in here. So, as you go along, there will be one concentration, then you can like say in terms of percentage, if I say 2% to say 25% or so, it could be different. What is done is then, so you can make it a step gradient, that is you can layer first 25%, the next one and the lightest one at the top, or it could be by a gradient maker, as we have discussed in earlier sections. By you can use a gradient maker to make a continuous gradient, where the lowest at the bottom of the tube contains highest gradient material, and the upper one or the starting of the tube has lighter or you can say lower concentration. The sample will be placed in here of a particular material or a particular say components, which could be macromolecules or subcellular organelles, and then centrifugation is performed. All right. So, what are the two methods actually which we were discussing about in density gradient centrifugation? One is called rate zonal separation. This is based on size of a particular analyte, and second method is isopenic or isodensity separation. Now, both of these methods are used for quantitative separation of components and determination of buoyant densities, and also for estimation of sedimentation coefficients that could be utilized. All right. So, let us start with first one, rate zonal separation. Now, these are based on differences in size, shape, density of the particle, density and viscosity of the medium, and applied centrifugal field. Now, rate zonal separation takes advantage of a particle size actually and mass instead of particle density. So, what is done here is for sedimentation of similar type of biological particles, which have almost like same density, but they have differences in their mass or size. Now, when you are preparing the density gradient in a tube, then in this particular method, maximum density of the gradient is chosen not to exceed that of the densest particle to be separated. So, this has to be understood that the maximum density of the gradient you have chosen in and you are putting it in the tube is such that it will not exceed the density of the densest particles in your sample. So, what does that means? That your sample largest particle can be pelleted in here, because the largest it is the largest density of the gradient is smaller than the densest particle of the sample. Now, Examples of common applications here include separation of cellular organelles, uh, particularly which has different sizes, then separation of proteins, uh, because proteins mostly have similar densities in a particular range, and but they have differing sizes actually. So, if you have 3 to 4 fold differences in their mass, then they could be separated on Reed zonal separation method. Now, sample is applied in a thin zone at the top of the centrifuge tube on a density gradient solution. So, what is done? First you have formed the density gradient in the tube, and once that is formed, it could be continuous or a stepwise gradient. Continuous is better always. Then sample will be applied on top of this density gradient. Then under centrifugal force, what will happen? The particles will begin sedimenting through the gradient in separation zones, that is spaced according to the relative velocities of the particle, and they will be moving based on their size, shape and density. Now, here function of gradient is mostly to stabilize the liquid column in the tube and to improve the resolution of the gradient. So, what is done is what is happening is here, they are mostly moving on the basis of size, and obviously the density, like certain components might have densities which are in the range of the density gradient. So, they will not move, but these are separating on the basis of size and density, but like many particles will move through, because densest part of the density gradient is smaller than the component materials here. Now, what is the criteria for successful separation here? So, what we have said, density of the sample solution must be less than that of the lowest density portion of the gradient. Now, density of the sample particle which we have taken, which is being separated, must be greater than that of the highest density portion of the gradient. So, the path length of the gradient must be sufficient for the separation to occur. Remember, if you so time is very important in here. So, because like if you perform the very long runs, that is if you perform a run beyond particular time, then the particles, which is densest particles, will certainly pellet at the bottom of the tube, and that will not be like they will not remain in the density gradient solution, and that will be a problem. So, what is done is that it is a time limited run has to be performed, that is you have to determine for how much time this run has to go. So, that to ensure that the particles do not pellet at the bottom of the tube. So, here it is shown very clearly that you have a density gradient, and you have this density gradient in here. You have a sample zone which is layered on top of the density gradient, and then centrifugation is performed for a particular period of time. Now, remember, this material should not be sedimenting or pelleting here, rather they should be separated in the column, liquid column only. So, as in a particular time when it is switched off, these particles are separated on the basis, mostly on the basis of their size actually. Like I said, these particles are mostly of same density kind. So, they will be separating on the basis of their size here. So, this is how rate zonal separation is achieved. So, what are the key features of rate zonal separation? In rate zonal separation, one is that it is like it is done for a particular period of time. It cannot be unlimited, it cannot be run for unlimited time. Then the density gradient which is formed, the densest part of the density gradient should be less than the densest component of the sample, so that pelleting does not take place. And as you do centrifugation for a limited time, these molecules will be separated on the basis of their size. If they are of the same density, but they are differing in 3 to 4 fold in their masses, then they could be separated. All right, let us move on to the next one, that is isopenic or isodensity centrifugation. Now, isodensity or isopenic centrifugation is depends upon the buoyant density of the particle, and it is not dependent on its size and shape here. So, it is a density which is the most important, it is exclusively depending on the density. Now, in this type of separation, a particle of a particular density will sink or will move during centrifugation until a position is reached where the density of the surrounding solution is exactly same as the density of the particle. So, once that is reached, the particle will be settled in that density or equal density. So, once this quasi equilibrium is reached, then the length of centrifugation does not really influence on the migration of the particle, because particle will not move further as it is floating on the cushion of a higher density material. So, these are very useful for separation of this particular method is very useful for separation of subcellular organelles, which are almost of the same size, but they differ in density actually. Like for example, you have like certain subcellular organelles like have 1.11 gram per centimeter cube, mitochondria, peroxisomes, they have 1.19 and 1.23 gram per centimeter cube density in sucrose solution. These could be all these subcellular organelles which have particular density, and they might be roughly equal in size. This method could be used for separation of these subcellular organelles. A common example for this method is the separation of nucleic acid in a cesium chloride gradient. Now, remember, so this is like one is you have layered the sample on top of the density gradient. So, then when you do centrifugation, it travels, the components travel and they settle down at their isodensity, and you can say they are floating on a cushion of higher density, because they cannot move further. You can run it for any time or at any centrifugal field, they will not move further, because the density of the gradient will be higher than their own density. So, they will just settle down where there is a equal density. Now, there could be another method where rather than overlaying the sample on top of the density gradient, the sample could be mixed in a particular density gradient material, and as the centrifugation goes, the density gradient material forms a gradient, and the components of the sample will automatically reach their isodensity. So, a very common method is, which is very well known, is the separation of nucleic acid in cesium chloride gradient. Now, these methods are a combination of sedimentation and flotation. It involves the layering sample not on the top of density gradient, but it is mixed in here. You can layer it also on top of that after making the gradient, like you have to run it for a long time to make cesium chloride gradient. These methods are commonly used in analytical centrifugation to determine buoyant density of a particle, base composition of double stranded DNA, and to separate linear forms of DNA. So, they could be, these methods could be utilized, this method could be utilized for determining buoyant density of the particle, and a lot of other like I said, the isopenic density gradient separation could be utilized for various application, like for separation of subcellular organelles, and for determining buoyant density, or for separation of DNA and other materials could be done on isodensity or isopenic separation method. So, we have done in here, we have discussed about two methods in density gradient centrifugation, that is reed zonal and isopenic. Both are different and they cater to particular needs. All right. Now, let us move on to the few more important points, like for example, what are the criteria for successful isopenic or isodensity separation. Now, in this particular method, density of the sample particle must fall within the limits of the gradient densities, like whatever gradient material you are utilizing, and the range of gradient you are making or forming, the sample density of the sample or the components of the sample should also be falling within those limits. Then here in isopenic any gradient length is acceptable, as it is isodensity or flotation method, there is no problem about the gradient length as long as the sample density falls within this range. Most of the time you can use the continuous or stepwise gradient, but it is preferable to use a continuous gradient for best results. The run time for isopenic separation must be sufficient for the particle to bend at their isodensity point or isopenic point. Excessive run times have no adverse effect, but if you run for a shorter period of time, then maybe the bending might not occur at the isodensity point. So, run time must be sufficient and that has to be determined. Now, these are so if we want to, like here it is explained in this figure on your screen. So, your material could be distributed or it could be layered on the top as you centrifuge, and as the gradient is formed, then different materials will be distributed according to their density. So, low density material will be at the top and very high density material will be at the bottom, and in between will be medium density samples. So, according to their density they will settle down at their isopenic or isodensity layer, and then they could be separated easily or recovered easily. Like I said, as an alternative to layering, these particle mixtures could be mixed with the gradient solution, and so there are two ways you can do it. Sample application, one is layering the particle mixture on the top of the preformed gradient, and another is mixing the material with gradient medium, and finally the gradient is self forming gradient materials are used like cesium chloride, and they will be separating, and this is called equilibrium isodensity method, as the material they will be forming the density gradient automatically as the centrifugation is performed, and also the samples to be purified will layer on their isodensity. Many salts of heavy metals like cesium or rubidium and other materials are utilized for forming the gradient for certain applications. Now, let us discuss some more points about the density gradient separations. One is formation and choice of density gradients. Now, all density gradient method involve a supporting column of liquid whose density increases towards the bottom of the centrifuge tube, and the function of density gradient here is, one is to stabilize the column of liquid in the centrifuge tube, to prevent mixing of the separated particles due to convection currents, to improve resolution of the separated components by eliminating factors such as mechanical vibrations and thermal gradients, and also density gradients. So, these are certain points which are very important, which you can say these are points or the function which determine the function of density gradient in these methods. Density gradient of different shapes can be produced, and like we were discussing, it could be discontinuous or step gradient, or it could be continuous gradient. Now, discontinuous or step gradient, it is done by overlayering method. So, what is done is known volumes of solutions of decreasing densities are allowed to run slowly down the side of the centrifuge tube to form layers over each other. It could be done by overlayering method or underlaying method, that is a layer of lightest density is introduced into the centrifuge tube with series of layers of increasing density placed over the first layer by pipetting. So, it could be both methods could be utilized. Discontinuous gradients can be used directly by layering of the sample to be separated as a narrow zone on the top of the layer, or if allowed to stand, layers slowly merge by diffusion to provide a continuous linear gradient. Many times what happens is these step gradient or discontinuous gradient, if allowed to merge, they will also merge and form a not a typical continuous, but sort of continuous linear gradient. They can be used for isopenic centrifugation, and these are suitable for separation of whole cells or subcellular organelles from plants or animal tissue homogenates, and for purification of say viruses or other components. So, there was discontinuous gradient, then continuous gradient. For continuous gradient making, you require a special apparatus called density gradient maker. Density gradient maker is capable of generating linear, concave or convex gradients, and these gradient makers consists of two chamber device for making continuous gradients. So, what you have is two identical chambers A and B connected by a particular tap channel contains equal volumes of the high density and low density material solutions respectively, and they the magnetic stirrer is there which keeps on mixing actually a particular chamber. So, the chamber B which sits on a magnetic stirrer bears a delivery tube which reaches to the bottom of the centrifuge tube via low pulsation peristaltic pump. So, when the labels of a liquid in the two chambers fall synchronously, dense solution from A mixes continuously with the higher solution in B, so that the pump delivers a solution of ever increasing density to the bottom of the centrifuge tube. So, the two stirring bars ensure that the label of the liquid in A and B is the same. So, if the two solutions have very different densities and if the stirring is not sufficiently vigorous, the dense solution may flow under the lighter solution. So, this is very important that mixing should be proper here. Now, if the placement of the two solutions is reversed, then the tendency of the less dense solution to float up through the denser solution in B will improve mixing considerably. So, in this format the tip of the delivery tube needs to be placed against the wall of the tube near its top actually. So, that a solution of decreasing density flows to the bottom. Now, this can be used for rate zonal or isopenic centrifugation, and different types of gradients can be made in here. Once the gradient is formed, then sample applications to the gradient has to be performed as we have seen. Now, optimum volume of the sample here and concentration should be determined, as each gradient has a defined sample capacity. The volume of the sample that can be applied is a function of the cross sectional area of the gradient exposed to the sample. So, sample volumes in the range of say 0.2 to 0.5 ml may be added to the say 1 to 1.6 centimeter diameter, and sample volumes up to 1 ml or 1 centimeter cube to tubes with diameter of 2.5 centimeter could be added. Effective separation could not be achieved with larger sample volumes. Remember, this is also very much important, that how much sample you are applying. For proteins and nucleic acids, sample concentration of say 1 microgram per ml to 1 milligram are recommended in a sucrose gradient. For rate zonal separations, samples should be applied with a narrow bore pipette slowly and gently to reduce mixing with the gradient. All right. So, so in this lecture we will continue our discussion in the next lecture about the formation and different aspects of the density gradient. So, in this lecture we have discussed about two methods. One is differential centrifugation and another is gradient centrifugation methods. In differential centrifugation method, it is the different sedimentation rate, that is at different applied centrifugal field particular components of the sample sediment, and that is taken, that criteria is taken, and increasing in steps the increasing centrifugal field is applied to sediment components at different at different centrifugal fields. In density gradient centrifugation, there are two methods we have discussed, rate zonal and the isopenic separations. In rate zonal separation, and mostly on the basis of size, like for example, many proteins with same density, but different sizes can be separated. In isodensity or isopenic separations, it is based on the density. So, many subcellular organelles having roughly same size, but different densities could be separated in here. In the next lecture, we will continue with the discussion of gradient formation and different points like recovery and monitoring of gradients, and also we are going to start with the rotors, different types of rotors which are used, and we will continue our discussion in the next lecture. Thank you.